It's a very hopeful time in myeloma because the field is changing so quickly. Not only are we getting new drugs and new options, but we're learning how to manipulate the immune system to attack the cancer for us. We've been doing this with stem cell transplants since the 80s, but we've been able to fine-tune it, and now really instead of using stem cells that require you reboot the entire immune system, we're focusing on T cells that are designated to recognize and attack the tumor itself. Specifically, the novel immunotherapies are focusing on CAR T cells, which are chimeric antigen T cells, where we modify members of the immune system, which are normal T cells. The process of producing CAR T cells starts with removing the T cells from the patient with a system similar to a dialysis machine, where the patient's blood is removed, run through a filter where the T cells can be extracted from the rest of the blood and taken into a lab. Those T cells are then engineered so that they are expressing the target receptor that we're looking for. At that time, they get expanded so that you then have a large number of cells, which can be reintroduced into the patient, and they then target and kill the tumor. Some of the problems with CAR T cells is that they may not focus on the tumor alone and may elicit a stronger immune response than we're expecting. This is something called cytokine release syndrome, which is one of the primary toxicities we are learning to mitigate. It will be really important to learn to treat the toxicity, which we have, but also to develop constructs that are less toxic, allowing maybe for us to move the therapy from hospitals into the clinic setting.